Ladies and gentlemen, in 1971, exactly 54 years ago, the very first edition of this contest took place. Back then, there were a little less than 30 contestants, but among them was one rather brilliant man, as we came to find out. He was soon to become an exceptional neuroscientist, an expert on Alzheimer's and Parkinson's disease. Not only did he become the head of Medical Research Council Lab of Molecular Biology at Cambridge, but he also won several prestigious distinctions, such as the Brain Prize in 2018, and of course a Royal Medal in 2019. I'd like you please to warmly welcome our keynote speaker today, Professor Michel Goedert.
Okay, thank you very much. Is so für dich die Purwörter Plätze bei ich auf Englisch am Schwenken. Vielmals merci für das Invitation. Und das Impressionante gesehen, wie viel Fondation Jonk Fuerscher zu meiner Zeit hat Jugend experimentiert seit 1971 geworden ist. Und ich werde über die Vergangenheit schwätzen, aber für dich wollte ich ein paar Worte über mich selber sagen. Und an der Gegenwart, nicht an der Vergangenheit. Und zum Schluss werde ich dann auf meine Arbeit angehen. Mein Lützeburg ist leider ein bisschen wackelig. And I therefore prefer to switch to English. The first slide. This slide shows you the second MRC, or Medical Research Council, Laboratory of Molecular Biology in Cambridge, and that's where I work. And in December, it will be 42 years since I began to work at the LMB. Now, for the first 30 years, I worked in this place here, which is the first MRC Laboratory of Molecular Biology, which is about 10 minutes away by foot from the second one. And for the past 12 years, we have been based in the second MRC Laboratory of Molecular Biology that was opened in May 2013. Now, the laboratory is not part of a university. The laboratory is a research institute that is funded by a block grant from the Medical Research Council. The Medical Research Council receives its money from the British taxpayer. It's something like the CNRS in France or the Max Planck Society in Germany. And in the current quinquennium, this block grant amounts to 238 million pounds, British pounds. And the LMB comprises something like 54 groups. Each group works on something completely different normally from the others. And each group consists of 10 or so people. Without there being individual budgets. So there's one big pot of money, and we all take from that. Some people call this a sort of a communist system, but it seems to work. And each group belongs to one of 4 divisions. So there are the cell biology, neurobiology, protein and nucleic acid chemistry, and structural biology. And I had one of those groups in the neurobiology division. We do not have to teach or to write grants, which is an advantage, so some people think we have cushy jobs. But our work is reviewed every 5 years, and we are scored between 1 and 10. 10 is the best, and 1 is the worst score. So we have to write about what we've done and what we want to do in future, and the score of less than 8 results in dismissal. So tenure, we're all tenured in paper. Tenure only means that you can work at the LMB for more than 6 years, basically, because every 5 years you have to go through this system. Now, the LMB is one of the best known research centers in the world, I would say. So members of its scientific staff have been awarded 12 Nobel Prizes between 1958 and 2018. And I've been working there since December 1983. Now, from 1954, when I was born, to 1973, I lived with my parents and two sisters in Avenue Gaston Diderich in Luxembourg City. And following the premier's examen at the Athénée, not far from here, I studied medicine at the University of Basel, where I obtained a medical degree in 1980. And this was followed by a PhD at Cambridge University, which I obtained in 1984. Now, by this point, I had decided to do research rather than practice medicine, which at that time, in a way, meant that I couldn't really come back here. I could have come back here, practice medicine, but doing research was not so easy. And to reach this decision, which was an important decision in my life, I was helped, I would say, by my taking part in these contests, what was then called Jugend experimentiert. So I took part in the contests of 1971 and 1972, and they were organized by Fernand Wagner, who was a teacher in chemistry at the Lycée des Arts et Métiers. And they were sponsored by Philips Luxembourg and the Luxemburger Wort. And here you can see Fernand Wagner with, alongside Grand-Duc Jean. Now, my 1972 project was on a form of plant cancer that is caused by a bacterium called Agrobacterium tumefaciens and gives rise to so-called crown gall tumors in plants. And I was awarded the first prize of that year's national contest, and this meant that I qualified to take part in the 5th European contest for young scientists and inventors organized by Philips, which was held at the Science Museum in London in 1973. And this contest pitted the winners of national contests against each other. So besides myself, there were people from Austria, Belgium, Denmark, Finland, France, Germany, Italy, the Netherlands, Norway, Spain, Switzerland, and the United Kingdom. And we had posters, a little bit like here, in front of which we talked to international judges about our work. And the chairman of the judges was the distinguished physicist Hendrik Casimir, who was also the head of research at Philips in Eindhoven. And I was fortunate to be awarded the first prize in the European Philips contest. So I received a diploma and a check over £500, which was quite a lot at the time. When my return flight with Luxair touched down at the Findel, so in May 1973, I was greeted by a welcoming party on the tarmac, the only time this has ever happened to me. And it was followed by a reception at what was then the Aero Golf Hotel. I think it's still there, but it has another name now. And here you can see me at this reception. So I was 19 years old at the time. And I'm surrounded by my father on the left, and then the man there who's laughing is André Heiderscheid. He was a director of the Luxemburger Wort, and I would say the éminence grise of the CSV and the Catholic Church. He was probably at the time one of the most powerful people in the country. There's Carlo Mainz, who was professeur attaché in the Service National de la Jeunesse. And then there is Marcel Varnier, who was the deputy head of Philips Luxembourg. Gesa Wertheim, who was the head of Philips Luxembourg, presented me with a cassette radio player that I still possess, but I must say it doesn't play music anymore. And at the time I was a student at the Athénée. It still looks exactly the same, I saw it before, where I did my premier's examen in the summer of 1973. 1973 also saw my first 3 publications. So the first in French is shown here. So it was in the Annales de microbiologie de l'Institut Pasteur. And note that my address is the Laboratoire de Biologie of the Athénée. The second publication was in Bild der Wissenschaft. Its founding editor was Heinz Haber. He was a well-known popularizer of science on television in West Germany in the 1960s and 1970s, and he had been a member of the jury of judges of the Phillips contest in London. Incidentally, this was also the first time that my name was misspelled in print, something that has happened many times since, especially in the United Kingdom, where my first name doesn't exist for a man, it only exists for women. And so even now, often when somebody calls me, they think I'm a woman before they've spoken to me. And so years later, it was revealed that Heinz Haber had been an ardent National Socialist in Germany in the 1930s and 1940s. And the third publication was in the Bulletin de la Société des Naturalistes Luxembourgeois. And my current total stands at 448 publications. And I now want to sort of turn to my ongoing work that began in 1987. I studied the filamentous inclusions that characterize many neurodegenerative diseases, including Alzheimer's and Parkinson's. And this is the list of diseases. Oh, sorry, this is the list of diseases that we work on. Oh, it's missing. All right, there should be a slide there with a list of diseases that we work on. So Alzheimer's disease, the most common, then there's Parkinson's disease, there's progressive supranuclear palsy, there's corticobasal degeneration, there is chronic traumatic encephalopathy. There's a total of 10 different diseases, and they're all characterized by the presence of abnormal filamentous inclusions in nerve cells, and for some of them also in glial cells. And these inclusions are made of tau protein, beta amyloid, and alpha-synuclein. And 6.5 of the diseases that should be there are so-called tauopathies. 3 of them are synucleinopathies, and only in Alzheimer's disease do you have both extracellular deposits of beta amyloid and intraneuronal tau inclusions. And there are no effective therapies for any of these diseases at present. And this slide basically sort of summarizes why one wants to study those filamentous inclusions. So this slide gives the sort of the main reasons for doing that. So there are 2 types of diseases. There are familial forms and sporadic forms. But they all share the presence of the same types of inclusions in the brain, in nerve cells, and sometimes outside brain cells. Now the sporadic ones are much more common than the familial ones. And the sporadic ones, basically, they happen to people in unpredictable ways. So some people that function, normally in an age-related manner, develop some of these, one of these, or sometimes even more than one of these diseases. Now, those sporadic ones are very difficult to study in terms of what causes them, because one could be anything, basically. But the familial ones are normally dominantly inherited. So that means that on average, 50% of somebody with disease will develop the same disease if they live long enough. And that allows you to find causes of disease. And this has been done. And the causes of disease are invariably in the genes encoding the abnormal filamentous inclusions or genes that influence their production. And that links inclusion body formation to the neurodegenerative process, and one then extrapolates from these familial cases to the more common sporadic ones. And one believes that the formation of these inclusions is what causes all cases of these diseases. Now, by this point, you may wonder what I have discovered in all these years, and this is sort of summarized here. So basically what we found is that tau and alpha-synuclein amyloid proteins are the central components of the filamentous inclusions that define Alzheimer's, Parkinson's, and other neurodegenerative diseases. And we've done a lot of work around this, but this is basically the major findings. And this slide shows you some of these diseases that we work on that contain tau inclusions. So you have Alzheimer's disease, tangle-only dementia, Pick's disease, chronic traumatic encephalopathy, progressive supranuclear palsy, corticobasal degeneration, and argiophilic brain disease. So they all have filamentous tau inclusions in their cells. And this is the diseases, so Parkinson's disease, dementia with Lewy bodies on the left here, and multiple system atrophy on the right. They are the so-called synucleinopathy. So this is basically what the main thing that we've discovered. Now this is all light microscopy. Now if you look by electron microscopy, you can see the abnormal filaments inside these inclusions. And then the question is, what do they look like, these filamentous inclusions? And this is something that we're still studying right now. Beginning in 2017, we've been using a technique called electron cryomicroscopy, also known as cryo-EM, to determine the atomic structures of tau, alpha-synuclein, amyloid beta, NTMEM 106B filaments, with some others as well, and other people in the LMB have determined the structures of filaments made of TDP43 and TAF15. And this summarizes, it just shows you the different shapes of these inclusions that we have determined. Now this slide looks to the future, and it says that we must be able to form the same filament structures under experimental conditions as those of filaments found in the human brains. And this is something that is not been possible. It's now possible in a limited way, but it's still not possible for most of these inclusions. So if you take just recombinantly expressed proteins, you assemble them into filaments, the structures of those filaments are not the same as what you find in the human brains. And we believe that this is necessary in order to detect those assemblies with great sensitivity and specificity, and also to study their formation, their spreading and toxicity, and most importantly, to design drugs that interfere with their formation. And in the past 3 years, we have been somewhat successful in this endeavor for some particular examples, but I'm not going to go into that. So I've now come sort of towards the end of my presentation, and I hope you can forgive me if I plug my book entitled Beginnings, that was published last December by Point Nemo Publishers. And you can purchase the book in the foyer if you're interested in learning more about the topics that I've been talking about. And I want to end by describing this painting, which is also on the cover of my book. It's by René Magritte, called L'Éternité. And I think it says something about science. So longevity of their published work is the prize researchers strive for the most. And along the same vein, it has been said that history is the ultimate judge of the relevance of a given piece of work. And scientific discoveries must pass the Magritte eternity test. And the painting shows 3 sculptures behind a velvet rope. There's the head of Jesus Christ on the left, the head of Dante Alighieri on the right, and a block of butter in the middle. And the truth of science must stand the test of time. I think that's the most important criterion, really, and not melt like butter. And then the question is, how do they compare to the truth of religion and poetry? And the traditional interpretation of l'éternité is that the truth of religion and poetry are eternal and that they will not melt like butter. An alternative view is that they are not eternal and that they will eventually melt like butter, which is not going to happen to the truth of science since they are immutable. Now, for every scientific problem, there is in general only one correct answer. Who finds it receives all the glory, with no place for any runners-up. And this makes scientific research a highly competitive undertaking. And this is something I think one has to be able to live with if one wants to do scientific research in a long time, because it happens once in a while that one works on something for a number of years and then somebody else publishes it somewhere else. but it basically means that one has wasted one's time in a way. So I think in scientific research, it's important to be able to live with things not working because other people are faster, also often experiments just not working. So most experiments don't work, really. And I think that's important if one wants to consider having a scientific career. I mean, on the plus side, it's never boring. There's always excitement, but it does take a lot of energy to do it. And I want to end by just giving an example of the longevity of published scientific work. So in 2013, when the LMB moved to its new building, on what is now the biomedical campus in Cambridge, we were asked by the City Council to name the street that the new building is located on after an alumnus of the laboratory. And we had quite a long discussion about this, and especially having seen several streets named after long-forgotten hospital administrators. The proposal in the end was to honor Francis Crick because his 1953 discovery with James Watson of the double helical structure of DNA and his subsequent work on the genetic code mean that his name will be remembered for centuries to come. And the MRC Laboratory of Molecular Biology is now located on Francis Crick Avenue. Thank you for your attention.